Okay, the way I said, those are the outline of what you're supposed to cover, and immediately if we are done with this one, we'll go to the next chapter. Now the nervous tissue, we are going to check on the overview of the nervous system, nerve cells, in particular the neurons. We are going to check on universal properties of these nerve cells, check on functional classes, structure of neuron, and finally we are going to do the axonal transport. Now, without wasting much time, we are just going to begin immediately. Then after we are done with this one, you're supposed to know that the next content to be covered, we are going to check on what we call the neuroglia, the supportive cells that are found within also the nervous system, the central nervous system. Okay, so immediately I'll first take you through the overview of the nervous system, and that is what is supposed to be done. So immediately we are going to start with the overview, and what you are able to see from your hand, that is the component of overview. That is the component of overview. Now we have the receptors, the neural integration, effector, central nervous system, peripheral nervous system, a nerve as large, a ganglion, sensory, that is efferent, and division or somatic. And then we are going to define each of them. That is the entire thing. Is when we now branch them into minor ones and again cover up. So the beginning of entire thing is that you need to know if the body right now is to maintain homeostasis and function of it effectively, the trillions of cells must work together in a coordinated fashion. Now, for example, now let's take if each cell behaved without regard to what others were doing, the result will be physiologically chaos and death, that is automatically. So this is prevented by two communication systems. And so in your body you have two communication systems. The nervous system, which is specialized for the rapid transmission of signal from cell to cell, and the endocrine system. I want you, in case you don't listen or get what I'm saying, just speak, voice yourself so that I can get you. Now I've said now the communication basis is done through the nervous system or the endocrine system. So the nervous system, it is specialized for the rapid transmission of signal from cell to cell, while the endocrine system is only specialized for sending chemicals message, the hormones that we know, through the blood. They can move through the blood like the endocrine, and then we can also have the exocrine, which just use the duct system. Now the most important aspect of both systems is that they detect changes in an organ, and after that they modify its physiology and modify that of other organ as well. And this one now, after this modification, this system functionally will now coordinate the organs of the body and play a central role in maintaining what we call the homeostasis. Now, if you are a scientist and you're well conversant with a lot of experimental and metabolism process, you also need to be aware that the scientific study of the nervous system is called neuroscience. And it is the same thing which now contain the neuroanatomy. So the study of structure, another side, we can call it neurophysiology. So I want you to know neuroscience as neuroanatomy and neurophysiology. I hope we are good up to there. And again, you need to know what is neurology. Neurology is also a branch of scientific study of nervous system, but now for neurology, the branch of medicine, it only deals with the diagnosis and treatment of neurological disorders. That is what we refer to as neurology. And in medicine, in clinical medicine fraternity, you do neurology, a portion of it in third year, and some portion also in first year. And again, you are supposed to do neurology in nursing as the third year. And the foremost people who are doing neurology in details are the MBCHB students, the medicine and surgery, the bachelor's ones. Now, we are going to check greater deal through this chapter up to almost third chapter, containing still the component of central nervous system. But why do we start with the nervous tissue? Because this chapter is the one which is primarily concerned with how individual nerve cells work. So the next 4 chapters are going to be concerned with the organization and function of the nervous system at the organ level up to the entire organism. And then we are going to also check the basic part of the nerve cell where now For instance, let's say some basic part of nerve cells, we are going to see them and see how they are introduced in the effectiveness of the entire thing. So before we go far, or maybe before I talk with of these items, I want you to confirm by viewing this image and see the segment of what is compounding the central nervous system and the entire nervous system. I hope you are able to see the image. Are you?
Yes.
Okay, thank you if you're able to see it. Now let's go back to the other slide. Okay, the first one is the receptor. So you'll need to know what is receptor, you need to know what is neural integration, you need to know what is effectors, you need to know what is central nervous system, peripheral nervous system, what is a nerve, a ganglion, a sensory division, all this one. That is what entails the overview of the nervous system. So the main objective around what we are talking about right now is that, first of all, at the end of this overview of the nervous system, you're supposed to be in a position to describe the major anatomical subdivision of the nervous system. Okay, just remember that what we are studying is so big and it has covered the whole body. That is from your brain, your spinal cord, and the entire projected body parts like your arm, your legs, the limbs, the limbic systems. Now, you have to be in position to start with dividing it at the major anatomical subdivision up to the minor one. That is objective number one after this session. Another one, you should be in position to state the general function of the nervous system and how these relate to the general classes of nerve cells. Another thing which you also should include is you are supposed to be in a position to describe the basic physiological properties of the nerve cells that enable them to carry out their function. So beginning from the receptor, the fundamental purpose of the nervous system is, number one, to receive information from receptor. So when you hear receptors, these are just but parts of the nervous system that are outwardly projected, and their main purpose is to detect any associated stimulus which can bring changes. And then after they received information from the system, that is the nervous cells, the receptor cells and the organs specialized to detect changes are very many in the body. So apart from the receptor, we still have other organs which are specialized to detect changes. So after they have received it, now they'll have to coordinate it. So, and its external environment also is a compounding factor, like the organism, what comes from the environment. Another thing, as a fundamental purpose of the nervous system, apart from receiving information from the receptor, is to process this information and determine the appropriate response. Now, if any step or if any a step called neural integration comes in now, if there is any information which has already been detected by receptor, we now end up having neural integration. That is to decide what step is the next to be taken. And then the third one which we have is effector. That is now the part that issues the command, what are we doing? We have already integrated the information which was received from the receptors. Now the effectors are gauging it and now giving it a command. So number 3 fundamental purpose is to issue commands to effectors. Example of effector is your brachial muscles when you want to remove your hands from a hot object. Right, so that is that. So mainly the thing that I wanted to know here that most body parts which play the role of effector are the muscles and the gland cells. The muscles and the gland cells. They're the ones that carry out the body response. Now I want you to see major anatomical subdivision which I was telling you. And according to you from your display, you'll only see them listed. So number 1, we have central nervous system and peripheral nervous system. Those are the main two divisions of CNS, of the entire nervous system. So according to your first topic in your the second semester in first years, that is in clinical medicine and surgery, you'll only be handling central nervous system and some compound of peripheral nervous system. But here we are going to evaluate up to the peripheral nervous system in much detail. Now, according to what you need to know about the central nervous system is that it consists of the brain and spinal cord, which are enclosed and protected by cranium and vertebral column. So the cranium protects the brain, and then the spinal cord is protected by vertebral column. And we have peripheral nervous system. From the word periphery. No, it means it's not within the center. It's within the adjacent boundary, outward in appearance, like peripheral environment, you see. So, being that the central nervous system just composed of the brain and the spine, which are in the center of the body, so your hands, your legs, they are the ones which now have the peripheral nervous system. And it consists of all the nervous system except the brain and spinal cord. So it is composed, this peripheral nervous system is composed of nerves and ganglia. So nerves, when I talk about nerve, you just see I've written there a nerve. So what is a nerve? Who can tell us what is a nerve? Any person can come in and tell us what is a nerve. We are still in the overview of the nervous system. Anybody with an idea what is a nerve? Who can tell us what is a nerve? Okay, nobody. So I'll have to proceed, right? Okay, a nerve. Simply, a nerve is a bundle of nerve fibers which is wrapped in fibrous connective tissue. Just a bundle of a nerve fiber. So I needed to ask me what is a nerve fibers. So we have that bundle which is wrapped, and then we have the nerve wrapped in fibrous connective tissue. That is what is a nerve. I'll talk about nerve at greater details in portion wise, but in later moments. So nerves emerge from the central nervous system through foramina of the skull. And when I say foramina, these are tiny holes, and I think you read them in skeletal system. Foramina of the skull and vertebral column. That is where they come. There are holes within the vertebral column or the skull, and now they come from those holes, and they carry signals. And when they carry signals and from other organs as well of the body, and then they relay it to where they're supposed to be taken. Another thing you can see from the overview is the word ganglion. What is a ganglion? A ganglion, which a plural term for ganglion is ganglia. Ganglion, ganglia. So ganglia is just a knot-like swelling in the nerve where the cell bodies of neurons are concentrated. Pale ziko nyingi. Cell bodies, I hope you know the cell body from your high school knowledge of response and coordination. Where they are accumulated, that is where we have the ganglia. There are too many cell body of neurons of those on those places. They are concentrated. That is what is forming a ganglion. And then another vital thing here is that you also need to know that the peripheral nervous system is also divided furtherly. So from there you see the division of the peripheral nervous system. So the peripheral nervous system is functionally divided into sensory and motor division. And each of these is further divided into somatic and visceral subdivision. So we have sensory and motor division. And the sensory part is divided into, the sensory part is divided into somatic and visceral subdivision. Then in motor division also is divided into somatic and visceral subdivision. So I just needed to play some trick when I show you this and try to rely on what I'm saying so that you can depict whatever you're able to see from your end. This image has it all. You can see from the top image we have central nervous system containing only the brain and the spinal cord. And then the peripheral nervous system we have the sensory division and the motor division. And each of them, starting from the sensory division of the peripheral nervous system, we still have visceral sensory division and somatic sensory division. On the other hand, on motor division, we have visceral motor division and somatic motor division. Now, on the motor division, the one with the visceral motor division is again divided. Visceral motor division is again divided into sympathetic division and parasympathetic division. And all these terms, you'll come to be familiar with them in a little while. So let's proceed. Now, before I project myself, I know people here, they have read and they're still reading, and so you can be able to distinguish what do you understand by the word sensory division and motor division. Just a question to pass by before we proceed. Anyone? Okay, no one, so let's proceed. Now, let us start with the sensory afferent division, because I've said in the peripheral nervous system we have two main division, the sensory one and the motor division. And then each of them is divided as either somatic and visceral. So let's start with the sensory division ones, and then see what happens. According to what you are seeing there will help you to relate what I'm saying. Let's see on the sensory division, and you can see where the sensory division is on the peripheral nervous system. As you can see, it is again divided into visceral sensory division and somatic sensory division. So what I need you to know that this sensory division, you can also call it afferent division. And when something is called afferent, it carries the information towards, like passing the information to afferent, like the afferent arterioles in the the nephron, right? So look at this. The sensory division of the peripheral nervous system carries sensory signal by way of afferent nerves fibers from the sensory receptors, which are located where? In your cells and the organs, right? Like on the skin. So they will detect stimuli, and then this sensory division will carry that information to the central nervous system. So it has to pass through the sensory division before reaching the central nervous system. That is why it is given the word sense. Sensory, sense, it senses. Now within the sensory division is where we have the two divisions, that is visceral sensory division and somatic sensory division. Now from the image you're able to see, now let's start by talking about visceral sensory division. The word visceral, the other time I told you, it has something to do with the organs. Can be the organs in your thorax, the organs in the peritoneum, the organs wherever. So the great thing here, I need you to know that the visceral sensory division carries signal mainly from the viscera, so from the organs. Can be the viscera of the thoracic, the way I've told you, abdominal cavity, such as the heart, the lung, the stomach, and the urinary bladder. On the other hand, somatic sensory division of the sensory division, that's another branch of the sensory division. It now carries signal from the receptor in the skin. The word somatic, I hope you remember in Form 3 when you are doing mitosis and meiosis 1 was taking place in somatic cells, right? So when I say somatic, I mean the normal cells like the skin, the muscles, the bones and joints, and not the internal organ. So the somatic sensory divisions relent information from those organs. Now let us go to the motor division. We are done with the sensory division of peripheral nervous system. This is just but an overview. We have not started detailing the entire thing. So away from that, the motor division, or what we call now motor efferent. So you can call motor division or motor efferent division. So that is that. They now carry signals by way of efferent nerve fibers. Now these nerves will come from the central nervous system to the effectors. So already sensory division has already taken the information from the viscera and the somatic region to the central nervous system. Now this central nervous system now relent the information to the visceras, to the effectors. The effectors are now the ones which are to be given command, the word to do, right? So when I say that, I mean, right? So the motor division will carry motor signal. Motor signal engage information to do with mechanical of action, the mechanism, like now moving, running away, withdrawing your hands from the hot thing, right? So like they will carry this information by the way of the efferent nerve. The fibers now will carry from the central nervous system to the effectors. Now the example of the effector I told you can be cells and organs that carry out the body's response, like mainly gland. And information from the central nervous system can go to your gland to inform it to release a specific hormone, right? So there are very many hormones which are regulating your body system. So glands play some of good roles or major roles or many roles when it comes to the to what we call the motor division. Now, in the motor division, we have what we call visceral motor division again, and we have somatic motor division. From what you are able to see, we want to talk about each of them. And we are going to start with visceral motor division. Now in visceral motor division, you can also call it autonomic nervous system. Visceral motor division can also be called as autonomic nervous system. And its main role is now to carry signals to glands. So the other one which we were talking about visceral, the other visceral which we were talking in somatic efferent division, when we were talking about the sensory division, The other visceral was removing information from organs. Now this one, in the motor's end, because this information is supposed to come from the central nervous system, it does not remove, but rather it now takes to the organs, or what we now can call as the autonomic nervous system. Now, before I proceed, any question from you guys before I proceed? Anywhere you've not understood so that I can clarify? Anywhere so that I can clarify? Are we good to go? I hope we are good to go.
Yes.
Okay, so if you are good to go, let's proceed. Okay, away from that, I was talking about the motor division of the peripheral nervous system, and I say that it is divided first into two, like just sensory division was divided into two, the visceral motor division and the somatic motor division. And I say that information of the motor division is carried from the central nervous system to the very effectors, the various effectors that are existing in your body. Can be gland, can be muscles, can be any cells, right? So I'd already talked about the motor division. Now I branch it to visceral motor division, which I was referring to as a fiber motion, or maybe a collective of nerves that were communicating to the organs, to the viscera. That is why it was given the word visceral motor division. Now, the visceral motor division, I've said also, it also can be called autonomic nervous system. Again, another important aspect here that I need you to know is that, what we call the visceral, the visceral motor division, it carries signals to glands, cardiac muscles, and smooth muscles. Not only glands, but also cardiac muscles and smooth muscles. So even that applies to autonomic nervous system, because autonomic nervous system is just the other name of visceral motor division. They play a vital role in glands, cardiac muscles, and smooth muscles, apart from the organs as large. So we usually have no voluntarily control over their effects. That's why even the cardiac muscles, it's not voluntary, it is involuntary, you see. So definitely we cannot control the autonomic nervous system or the visceral motor division. Now, and this system will operate at an unconscious level, whether you are sleeping or awake, whether you faint or you are in coma. Most organs will still work alone. So that is why we call them autonomic nervous system. So the response of this system and its effectors are what we call the visceral reflexes. The visceral reflexes. I want you to remember that term and you'll check it later. So then the autonomic nervous system has two further divisions. The autonomic or the visceral motor division has got two more, and you are able to see it from your hand. The sympathetic division and parasympathetic division. Now, beginning from the sympathetic division, it tends to arouse the body for action, right? It stimulates the body for action. That is what I mean. It arouses the body for action. So don't forget that. Its main function is to stimulate the body. That is sympathetic nervous system. The sympathetic division, So look at this. After this, I need you to know, for example, when you are by accelerating the heartbeat, you see, I talk about the effect of autonomic nervous system, the visceral motor division on the cardiac muscle. So, for example, by accelerating the heartbeat and increasing respiratory airflow, that is the work of autonomic nervous system of sympathetic division. They'll always tend to increase. They pace it up, zinaongeza. And then on the parasympathetic division, I need you to know that it tends to have a calming effect. It reduces, right? It reduces. So it slows down the heartbeat, for example. But now, you will get that, for example, when sympathetic division does the opposite, kama in some organ, not all the organs sympathetic division will do the arousal. Though in majority of the organs, sympathetic division zitakuwa zina stimulate. But, for example, if you compare the effect of sympathetic division of the nerves in the digestion system and the one which is in the cardiac system, cardiovascular system, you realize that in digestion, instead of it arousing, it inhibits. And then parasympathetic division, in digestion, instead of it calming down, it stimulates. We are done with the visceral motor division. I'm talking finally about somatic motor division, and then we are going to be done with the overview. So after overview, we are going to check the nerve cell one by one, the neuron. The nerve cell is the other name of neuron. So let me talk about the somatic motor division. The somatic motor division. Now, like I said the other time, it carries signals to the skeletal muscles, and this output produces muscular contractions that are under voluntary control. So they take information to the skeletal muscles, as we all know, and now these muscles, they are under voluntary control, unlike the other one, which was played by autonomic nervous system or the visceral nervous system. Now in this output, they will produce muscular contraction, and so they are under voluntary control, as well as involuntary muscle contraction can also come. So both of them can come into play. Some muscles will display the voluntary, some will be involuntary, but they are still under somatic motor division. And what we are getting from this activity is what we refer to as the somatic reflexes. So foregoing and all this, the foregoing terminology may give the impression that the body has several nervous systems: the central, peripheral, the sensory, the motor, the somatic, and the visceral. These are just terms of convenience. But however, I need you to know there's only one nervous system, and these subsystems are interconnected part of the whole. I hope we are together up to there. And kindly just mute your microphone if you've opened it, because you are producing echo. Okay, thank you. So let's proceed. Are we good up to there?
Yes.
Okay, let's produce. Thank you. Faith Murungi, thank you. So let's proceed. And now, another important thing here now you're going to have is the nerve cells. And from your hand, you can see the component of what you're going to have right now. In the nerve cells, we're going to see the universal property. We are going to see the universal properties of a nerve cells, compounding, the excitability, conductivity, and secretion. Those are the key things we are going to start by. And immediately after that, we are going to check what is next. But here we start. So on the nervous cells, I want you to know that there are objectives that we are going to outline before we proceed. And first and foremost, you need to know that you are supposed to be able to identify the parts of a neuron after this slide. Again, you're also supposed to explain how neurons transport materials between the cell body and tips of the axon. That is just but a part of the neuron, so don't be scared. So you see how they move, the information moves within a neuron. It's also supposed to be able to name the cells that aid neuron function and state their functions as well. You'll be expected to describe the myelin sheath that is formed around certain nerve fibers, just the way you did in high school, right? And you're also supposed to be in position to explain its importance. And you're supposed to explain how danger or damaged nerve fibers can regenerate. So along the nerve cell, those are the component or outline you're supposed to be aware of after this. So let's begin by the first subtopic, that is universal properties. And here I want to talk about excitability, conductivity, and secretion. And I want to say today's class is so dormant, and please just make it lively by answering the following question according to your own understanding. So what can you say about excitability of a nerve cell? Let me start with Faith Muriungi. Then Faith Muriungi, what can you say about excitability? Then you go to Florence Ngina, Karuma Brenda, Simon Ekal, Nelet. Yeah. Somebody somewhere? Anybody who can say anything about excitability, conductivity? Today we don't have Nicole. Okay, but you're great. So let me just proceed. Now, when you talk about excitability, in high school we used to call it irritability. Just ability of an organism to detect changes in the environment, that is stimuli, and then they respond. They get to be stimulated by those changes. Now, excitability, all cells possess excitability and the ability to respond to the environmental changes. And these changes is what we call the stimuli. So stimuli can be fire, can be kiboko, can be rain. You see, there are very many types of stimuli, right? Somebody want to join? Let me add person. Oh, he has gone already. Okay, so I'll say there are many stimuli. So neurons have developed this property to the highest degree. So that is one universal property of a neuron, excitability. And then we have conductivity. Conductivity, these neurons, they respond to stimuli by producing traveling electrical signal that quickly reach other cells at a distance location. So that conduction is what we call conductivity. We still have secretion as another property. When the electrical signals now reaches the end of the nerve fiber, mwisho yake, just like you did about in high school, the neuron will secrete a chemical neurotransmitter that jumps the gap and stimulates the next gap or the next cell. So what I want you to know, I just want you to think about this. What basic physiological properties do a nerve cell and skeletal muscle fiber have in common? I know we have not done about the skeletal muscle fiber, and we'll do it if the time is there. So what basic physiological properties do a nerve cell and skeletal muscle fiber have in common? Another thing that I need you to think about is that you need to be in a position to name a physiological property of each that other one lacks. Like, let's say, what do we have in nerve cell that skeletal muscle do not have? So that is that. So I've talked about excitability, conductivity, and secretion. We are going to the next slide. And in that slide, we have what is called functional classes. So you are able to see the functional classes. Now, on the functional classes, we're going to see classification of the neurons based on their function. The sensory neurons, or what we call the afferent ones, the interneurons, what we call associated neurons, and the motor or efferent neurons. So we're going to start with the sensory neuron. But before we start, I just want to tell you something. There are three general classes of neurons that you need to know. One is, they correspond to the three major aspects of nervous system, what we had already talked about earlier. So the way we have three, and you also remember we talked about three aspects of nervous system, right? The receptor, the integral, where the integration was taking place, that is the neural integral, and then we had the effectors. Now, talking about this, we are going to start by the sensory neuron, so what we refer to as the afferent neuron. I said afferent, it carries two. Efferent carries away, that is the word. Now let's start with the sensory of the afferent neurons. They are specialized to detect stimuli such as light, heat, pressure, and chemicals. And another thing is that they will transmit this information about them to the central nervous system, or CNS. And these neurons can begin in almost any organ of the body and end in the central nervous system. So the word afferent, what I was telling you, it refers to the signal conduction toward the central nervous system. So some sensory receptors such as pain and small receptors are themselves can act as neurons. So we have pain receptors, they can as well act as a receptor or neurons, so they can communicate without requiring even the axons. So in other cases, when you talk about the taste and hearing, the receptor is a separate cell. It's a separate cell that communicates directly with sensory neuron. So some of the small receptors, they'll act as the neuron and the receptor. But in some, we'll have a receptor away from the neuron body. Talking about the interneuron, still on functional classes, let's talk about interneuron or associated neurons. These interneurons are what we refer to as the relay neuron in your high school syllabus. They lie entirely within the central nervous system. So they receive signals from many other neurons and carry out the integrative function of the nervous system. That is, they will process, store, and retrieve information. They process, they will store, and they will retrieve the information. And then they'll decide, they make decisions that determine how the body will now respond to the stimuli. So roughly, let's say around 90% of the neurons in your body, our neurons, they are interneurons. They are located within the brain and the spinal cord. Those are the central nervous system. So the word interneuron refers to the fact that they lie between those two segments. So that is very important, and I'll show you a certain image here. Before I show you that, I need you to know that they will lie between and they will interconnect the incoming sensory pathways and the outgoing motor pathways of the central nervous system. That is why they are called the interneuron. Away from that, let's talk about the motor neurons. The motor neurons, they send signal predominantly to muscles and glands. So to mean they normally play the major role on muscles and glands. So they are like stimulating them. So they send signal from the central nervous system to the muscles and the glands, and the effectors that carry out the body responses to the stimuli. So these neurons, we call them motor neurons, right? Because most of them, they lead to muscle cells. That is why we call them the title motor neurons. And they can lead to either muscle cells or the efferent neurons to signify the signal conduction away from the central nervous system. That is all about that, functional classes. So away from that, I wanted to be able to see the image I've just shown you there. Yeah, you can see we have sensory afferent neurons. Are you able to see? Is it displayed? Are you able to see? Can somebody say anything?
Yes.
Okay, if you're able to see. So in one, you can see we have sensory neurons conducting signal from the receptors. Let's say that is sweat nerves, or let's say just in the skin, the epidermal nerve endings, right? So they move the information to the central nervous system, can be in the brain or spinal cord. They find interneurons that now make decision, and then they confine the information in the central nervous system. Then they move it through the motor neurons to the, let's say, muscle cells, right, or the glands. That is that. Now you can come back and replay this thing in YouTube as many a time you want using our YouTube channel, so that at least you can be informed in a broader detail and remember what we are discussing. If there is any question, and if there is no, let us go to structure of a neuron. What you're able to see from your end is the structure of a neuron. And the component of a neuron, you see we have soma or cell body, or what we can call the pericardium. I've given even the pronunciation of the name so that you don't make mistake. We still have neurofibrils, the dendrites, the axon hillock, the axon or the nerve fibers. We have the axoplasm, the axolemma, terminal abrasion, synaptic knob, the synapse, and the synaptic vessels. Now we are going to talk about all these ones, and you're going to see in the image and diagram so that you can have a distinction when you are distinguishing them. Now let's start. There are several varieties of neurons, as well as we are going to see them in details. But a good starting point for this discussion is that neuronal structure is a motor neuron of the spinal cord. So we are going to see neuronal structure based on the component of motor neuron of the spinal cord. That is the one which is normally used, so that it can give you a diversified and a significant detail. So this one, they control the center of the neuron, and the center of the neuron which control the neuron, or what we call the control center of a neuron, is what we call the soma, or you can also call it the cell body or the pericardium. It has a single centrally located nucleus, and I am sure you are informed of that from your high school. And then it has a large nucleolus, very large. And then the cytoplasm contains mitochondrion, which is important for energy. I hope you know that. It also has lysosome. It has what else? It has Golgi complex, and very many inclusions and extensive rough endoplasmic reticulum which help it in transmission of secretion. And also, you know, every cell must have cytoskeleton for integrity. So cytoskeleton, I know you know about cytoskeleton, you did it in your first semester, I'm very sure. Now let's proceed. So the cytoskeleton consists of a dense mesh of microtubules, and you know about the microtubules, the neurofibrils, and that is where the word neurofibril comes in. So the bundles of actin filament that compartmentalizes the rough endoplasmic reticulum, or this is what, or into it, make it appears like dark stained regions. So that is what we call the neurofibril, the bundles of actin filament. They compartmentalize, like they give it a compiled docket, make the rough endoplasmic reticulum appear like dark stained region, and this one is what we call the NISI. If you go and check the NISL, you see the NISL bodies. The NISL bodies are unique to neurons. Each neuron has got only its NISL bodies. They are unique to each neurons. That is what I need you to know, and they are very helpful clue to identify a neuron. So they help in identifying them in tissue secretions. And when you do histological sections, the one I told you when you're preparing temporary slide, and you have mixed cell types, what you'll use to identify specific tissue or nerve cells is what we refer to as the NISL bodies, because they are unique to each neuron. And again, mature neurons lack centrals and apparently undergo no further mitosis. After they mature, they don't have central. And as you can remember in your high school knowledge, central was very important in cell division, especially when you're talking about the mitosis and meiosis, not the other division like vegetative propagation and budding and binary fission, the ones you did. So here, the central are lacking after they are mature, and therefore they will not undergo mitosis. So ukiumia, when they are mature, they will not develop again, and that is what we refer to. You see people having brachial palsy. You see, definitely if some nerves in your body are destroyed, especially when you are old, they will not regenerate. So like a case of accident, a trauma that will interfere with your nerves, maybe the radial nerve, it can cause your hand to drop, not because of it can't, or the doctors can't, but because it cannot regenerate. You've already tampered with the centrals which are already there, and you don't have any central being found in this region. So that is that. So I need you to know they are unusually long-lived cells, the mature ones, and they are capable of functioning over 100 years. Even into old age, you can still use your neurons. But we have a category which is exceptional. They are unspecial. When you are old age, the nerve fibers and the cells, there are unspecialized stem cells in them, in the central nervous system, that can divide and develop into new neurons. So look, on your body, the body part, you can, at most, you can injure a nerve fibers which is not old and it will regenerate. But in your brain, they are unspecialized for that division. The stem cells which is there cannot regenerate them, whether they are mature or immature. If you cause any destruction in your brain content, it goes and never come back. There is no regeneration there. Another information that I need you to know that the major cytoplasmic inclusion in the neuron are the glycogen granules and lipid droplets and melanin. And another one which is not always being used is what we call the golden brown pigment, the lipofuscin. I know you've known lipofuscin for a long time in your biochemistry knowledge. That is an end product of lysosomal digestion of worn-out organelles and other products. So lipofuscin collects out with age and pushes the nucleus to one side of the cell. And the same lipofuscin granules are also called the wear and tear granules. So why were they called wear and tear granules? Because they are most abundant in old neurons, but they are apparently harmless at all. So still on the soma and the neurofibrils. The sum of the neuron usually give rise to a few thick processes, what we now call the dendrites. So they branch into vast number of dendrites. So those dendrites are what are called the processes that originate from the cell body or the soma, because I told you that soma is also referred to as the cell body. So dendrites, where is the name dendrites coming from? Who can tell us where the name dendrites comes from? you know, history of medicine is so important and it's good. So dendrites, they were named from their striking resemblance to the bare branches of a tree in winter. So the winter tree normally has some branches, numerous bare branches. So the dendrites got the name from there. So the dendrites are the primary site for receiving signals from other neurons. So like adjacent neurons can communicate using the dendrites. And then another thing I needed to know that some neurons have only one dendrite and some have thousands. So the more the dendrites a neuron has, the more information it can receive from the cell and incorporate into its decision. So don't forget that. Now the image below, I don't know if you're able to see that image. Are you able to see any image? Are you? Are you able to see any image?
No.
Now you are able, right?
Now you are right.
Okay.
Yes.
Okay, now, yeah, you see that image. You see that image? And there's another images I'll show you. So those are the dendrites I was talking about. So I can see somebody want to join. Let me admit that person before we proceed. Several people. And they keep on coming and going. So that is that. I'd said initially before reaching here that the more the dendrite, the more the information is being collected, so easy decision making. That is that. Other thing is that, yeah, there are some tangled. As the dendrites may seem, they are looking like tangle, but they provide what we call exquisitive or what we can call compressive or precise pathways for the reception and processing of the neuron information. So on one side of the soma, we have a mold called the axon hillock. From your hand, you can see axon hillock. If you're able to see my cursor, that is axon hillock. So that axon hillock, it is where the axon or the nerve fiber originates. So that is like it produces the nerve fibers. And then the axon is cylindrical and relatively unbranched for most of its length. So although it may give rise to a few branches called the axon collaterals along the way, but most axons will branch rapidly and they will branch extensively and at their distal end. An axon is normally specialized for what we have or what we refer to as the rapid conduction of nerve signals to points remote from the soma, or maybe they are more flexible than the entire thing. So you can find zingine ziko refu. Some are as long as 1 meter, yeah, you see. So they can reach the interior and remote area in your body. So the cytoplasm, its cytoplasm is called the axoplasm. The cytoplasm of the axon is called the axoplasm, and you can see it there. And it is membrane which surrounds is what we call the axolemma, the membrane that bound the cytoplasm of the axon or the axoplasm. It's called the axolemma. And then we have a neuron nerve. It has got more than let's say one axon, and some neurons may have only one or none. Like for example, in your retina, in the retina, the neurons which are found in the retina and the brain, they have none of axon. So they can range from 5 to 135 micrometer in diameter, while axon can range from 1 to 20 micrometer in diameter and from a few millimeters to more than 1 meter long. Again, so for all these dimensions I've been mentioning, such a dimensions, right? Such a dimensions, let's say, are more impressive when we scale them up to the size of the familiar object. For example, if the sum of our spinal motor neurons were the size of a tennis ball, right? Its dendrites will form a huge bushy mass that could fill a 30-seat classroom from floor to ceiling. So its axon will be up to a mile long, but a little narrower than a garden hose. So this is quite a point to ponder, right? The neuron must assemble molecules and organelles in it. So the tennis ball, that is the soma, and the liver. So when the neuron is big, it has to have some proportion with the soma, the cell body. It has to be in a position to assemble all the components and push inside the cell body, the soma. That is why I've given you example of the classroom. So it has to compile this information and deliver them through its mile-long garden hose to the end of the axon. So how it achieved this remarkable feat is unexplained, and we are going to explain it shortly later. So we'll talk about this one into details, but let's go back to the other slide. I hope you've seen that image for long. Now let's use this outline so that I can see what I'm talking about in the next. I don't jump, right? So we have already talked about axolemma, axoplasm. Now we are going to what is referred to as the terminal arborization. So at the distal end of axon usually have what we call terminal arborization. And I'm going to show you an image of this. It's an extensive complex of fine branches, and you're going to see it how it looks like. So the synaptic knob is the next thing. Synaptic knob or terminal button. I'm going to say about this one, but I am very sure that you did it in high school. It's like a little swelling that forms a junction, and that junction is what we call the synapse. So between one nerve to another, we have a junction called synapse, and each end of the synapse we have synaptic knob. Just simple like that. So with a muscle cell, for example, gland cells or another neuron, we can still have a synapse. So not only from one nerve to another, but even to the muscle cells and other neurons, and also even the gland cells. And within the synapse or the synaptic knob, it contains what we call the synaptic vesicles, which is full of neurotransmitters. And I'm sure up to now you are informed of what is a neurotransmitter. Now, not all neurons will fit in the preceding description. Some neurons are classified structurally, right? And we are going to see that one. But before we go into that details, I want to show you the image so that you can see the arborization. But I am sure that you should have seen it in the previous image. Yeah, this place here. If you are able to see what I'm pointing, are you able to see my cursor? Yeah, that is what we call terminal arborization. Okay, that is that. Let's proceed. Okay, we are going to see how neurons can be classified. I've said not all neurons have got these details, but there's some which are very funny based on the axon, the number of axons. So that is what we are going to have next, and you're able to see it from wherever you are. We are going to see classification of this neuron. So we have structure of neuron. Neurons are classified structurally according to the number of processes extending from the soma. We have multipolar neurons, we have the bipolar neurons, the unipolar neurons, and the anaxonic neurons. So if I was able to inquire, I could have asked, who can tell us anything about multipolar, bipolar, unipolar, and anaxonic neurons? Any among, you can say any of them, so that we can save my time. Tell us anything. Okay, let us save time. I'll just proceed. Mm-hmm. Mm-hmm. Wow, so it processes a thing. It consists of what? A single axon and many dendrites, right? What about the bipolar neurons? Okay, you see what are all these in a little while. So we have multipolar neurons. They are those like the preceding that have one axon and multiple dendrites, the way he has said it, right? And this is the most common type of the neurons that include most neurons, the brain, and the spinal cord. And again, we have bipolar neuron. What about bipolar? Can anybody say anything about bipolar neuron? Yeah, it has got one axon and one dendrite, correct? So they have one axon and one dendrite. Example is olfactory cells of your nose, the nasal cavity, and of the retina. Some of the retina neurons also have that one. And inside the ear, the others also which are bipolar. Still on the same. What about unipolar? Unipolar neuron. These ones, they have only a single process leading away from the soma, right? Only a single. So they are represented by the neurons that carry sensory signal to the spinal cord. These neurons can also be called the pseudo-unipolar. Because, why do you think they can be called the pseudo-unipolar? It's because they start out as bipolar neurons in the embryo, but their two processes fuses into one as the neuron grow and develop. That is why they can as well be referred to as the pseudo-unipolar neuron. So I want to show you an image here before we proceed talk about all this. But you need to also know that when you talk about unipolar, a shorter distance from the soma, the process branch like a T with what we call the peripheral fiber. So they carrying signal from the source of the sensation and central fiber continuing into the spinal cord. Now in most other neurons, a dendrites carry signal toward a soma. Inatembea ikienda kwa soma, right? And carries nerve signals directly to the spinal cord. Now the dendrites are branching, that is in these unipolar neurons. Here the dendrites are the branch, and they are branching receptive endings, and the skins, for example, and other places of origin, they'll move. So like, while the rest of the fiber is considered to be the axon, right, the way they are defined in these neurons by the presence of the myelia and the ability to regenerate action potential, we are going to see later on how these pseudo-unipolar are coming into existing, both structurally and functionally. Axonic or anaxonic neurons, from the word ana, haina, anaxonic neurons, huyu ana axon. So let's see what about anaxonic neurons. So like, they have multiple dendrites, but they don't have any axons. They communicate through their dendrites and do not produce action potential. So some example of anaxonic neurons are found in the brain, some are in retina, yeah, like in retina, they normally help in visual process such as the perception of contrast, how they can see deviation, the contrast, the blur, the blah blah blah, that is. So the next nini we'll be talking about the axonal transport, and that is the last part of this lesson. And before then, I'd like you to see this image so that you can see how they look like. I hope you are able to see what is there. We have the dendrites. On the first is multipolar neurons, second one is bipolar neurons, unipolar neurons, and an axonic neuron. So let's go away from this and do the axonal transport. This is the last bit of this content. Now starting from the axonal transport, we're still going to talk about the anterograde transport and retrograde transport. Now on axonal transport, all of the proteins needed by the neurons must be made in the soma, that in the cell body, where the protein now synthesizes, helps process into the organelles such as the nucleus, the ribosomes, and the right endoplasmic reticulum, or where they are located. Now, yet many of these proteins, yeah, they are needed in the axon. For example, so they are being synthesized in the soma, but they are needed in the axon. For example, they are needed in the axon to repair and maintain the exolemma, to furnish the ion gates. I know you know about. No, you are going to do the ion gate for clinical medicine the next semester. They are supposed to furnish the ion gates in the membrane, or to act in the synaptic knob as an enzyme and signal molecules. So they have to move from soma to the axons. So the other substances are transported from the axon terminals back to the soma for disposal or recycling. The two-way passage of proteins, organelles, and other materials along the axon is what now we call the axonal transport. How they move, that is what we call the axonal transport. And this movement from the soma down to the axon is anterograde transport. And the movement up from the axon toward the soma is called the retrograde transport. So we have the anterograde and the retrograde transport. So materials which are living or they are traveling along the microtubule, the cytoskeleton, which act like a railroad, njia ya gari ya moshi. So it guides them to their destination. But when is the motor, in the motor now, the motor yani the mechanism, what is the motor that drives them along the tracks? What do you think? Instantly, the anterograde transport. It employs a motor protein called the kinesin, and you'll see this one in later. Then the retrograde, it uses what we call, and we are very familiar with, the dynein. The dynein, right? The dynein, or the same protein we encountered earlier when we are talking about cilia and flagella in the structure of the cytoskeleton. So these proteins carry materials on their backs, and they reach out like the I don't know how I can put it, like the myosin head of the muscle. When you check on the muscle contraction, they try to bind repeatedly to the microtubule and crawl along them. So there are two types of axonal transport, fast and slow. So we have fast axonal transport and slow axonal transport. And when we are done with that one, the entire nervous tissue is done. So the next one we're going to do is supportive cell, the neuroglial, which is maybe late at night. So that is that. So I want to talk about this type of two axonal transport, the first one and the slow one. So starting the first axonal transport, it occurs at the rate is always 20 to 400 millimeters per day, and maybe either anterograde or retrograde. That is the first one. So let's start with the first anterograde, then check on the first retrograde. In the first anterograde, transport move mitochondria, synaptic vessel, all those other organelles, yeah, their component. They move, and then the components of the axolemma, calcium ion, zyme channels such as the acetylcholinesterase and some molecules, they move them. They move them from the soma to the axon. Now, on the first retrograde transport, it returns now the used compound, those items that you were transporting. Now, after they are used, it is taken back, or they are taken back to the cell body or the soma. So the first retrograde, they return using synaptic vesicles and the materials to the soma, and they inform the soma of condition at the axon terminals. And then some pathogen exploit retrograde transport to invade neurons, and when that one happens, we'll have the ugonjwa kama tetanus. So the toxins of the tetanus, yeah, they can invade the neurons and they affect the retrograde, right? Like the herpes simplex, which is STI, and also comes as a result of HIV infection. So by this, there will be delay between. Okay, I want you to understand some concept here. So tetanus, the herpes simplex, the rabies, the polio virus, all those ones, they can affect the nervous system. So in such infection, there will be delay between infection and the onset of symptoms, corresponding to the time needed for the pathogens to reach the somas. Why do they delay? Because they have already interfered with the transmission. So there will be delay. Now, away from the fast transportation or fast axonal transportation, let's talk about slow axonal transportation. This is also called axoplasmic flow, and it occurs at a rate of 0.5 to 10 millimeter a day. And again, I need you to know it's always anterograde only, but not retrograde. So in a term, it can move enzymes, cytoskeletal component down the axons. It can renew worn out axoplasmic components in mature neurons and supplies new exoplasma for developing or regenerating neurons. Damaged nerve cells can damage the ones which are damaged. Yeah, the damaged nerve fibers, they can regenerate at a speed governed by the slow axonal transport. So before we go away and we end this class, if you go, I want you to check to answer the following. You need to be in a position to sketch multipolar neuron and label the soma, dendrite, axon, and terminal labialization, synaptic knob, myelin sheath, and the nodes of Ranvier, which I have not told you. I'm also going to share for you this entire slide in your WhatsApp, but late at night when I'm free. And again, I need you also to be in position to explain the differences between sensory neuron, motor neuron, and interneuron. You also need to know what is the functional difference between dendrites and axon. You also need to be able to answer how the proteins and the other chemicals synthesized in the soma can get to the synaptic knob. By what process can a virus invade the peripheral nerve fibers? How can they get to the soma? What process that is? It anterograde or retrograde? So the next chapter of the nervous tissue we are going to see is on supportive cell. Thank you for your time. Thank you for being here. Courtesy of Medical Students Arena, myself, Amuma. Have a nice time. Leban Jiru, you are joining when you are leaving.